Welcome back to the discussion of membrane transport. For the passive processes, we are looking at facilitated diffusion, the third type of mechanism of passive transport. We've already by now have discussed simple diffusion by random movement driven by concentration gradient. We've talked about osmosis already, which is solvent movement, and it's driven by differences in solute concentration, driven by tonicity, driven by osmotic pressure. So facilitated diffusion, yes, there is no energy that is expended, except this type of transportation is aided. There is help provided not by simple random movement of the particles, but it is because the particles are actually either bound by the carriers and shuttled across the membrane, or the channels actually bind them and help in their transportation across the cell membrane. So this is facilitated diffusion, and as a result, it will be different from simple diffusion in as far as the actual mechanism of transportation is concerned. And so we can see the differences that facilitated diffusion is specific for particular chemical substances. Why? Because there are particular channels or carrier transporters for certain ions or molecules only. So they are very specific. If it's a carrier for amino acids, it's a carrier for amino acid. It will not be able to transport carbon dioxide, for example. No. If it is a pump, if it is a carrier really for let's say an ion like bicarbonate, it is a carrier for bicarbonate, nothing else. Then this is also similar with a random movement in simple diffusion, that it is passive. There is no energy that is used. What directs the movement is the concentration gradient, but there is help of carriers and channels. Also, the other difference with a simple diffusion and osmosis is that facilitated diffusion is saturable. It reaches a limit beyond which it cannot increase the transportation. Yes, there is a concentration difference, but here, if the carrier is carried enough and all the binding sites are filled, no matter how much the concentration difference is, the addition of solute cannot increase the facilitated diffusion of the substance. Let's move now to active transport, the mechanisms of transportation that needs energy. So the energy is in form of our favorite molecule called ATP, adenosine triphosphate, and here chemical substances are being transported from lower concentration to higher concentration, meaning they are being concentrated in one side of the cell. And so here also we want to mention that the driving force is ATP, not the concentration difference, but ATP. Yes, there is requirement for use of carrier proteins as well as pumps. The two main types of active transport, especially for small molecules, is primary active transport, which uses membrane pumps, or secondary active transport, which is referred also as co-transportation, which is a co-transportation. So membrane pumps, which use primary active transport, use ATP to move a chemical substance across the membrane up the concentration gradient. So we have an illustration here. From outside the cell into the cell, there is this carrier protein that binds the chemical substance or the solute. The binding of the chemical substance activates the conversion of ATP to ADP, and the breaking of those bonds is what drives the transportation. Then we have another example of active transport, produced by the sodium-potassium pump. This is a very important pump in our body because it is found in all cells, so it is said to be ubiquitous. It is very, very important in creating the concentration gradient for sodium and potassium. Very, very important for the body. And because it pumps a lot of sodium out of the cell and pumps less potassium into the cell, it ensures that inside the cell there is less solutes than outside. So it maintains the cell volume. So the cell volume for all our cells is dependent on the function of the sodium-potassium pump. Without it, if it fails, all our cells will be in problems because there will be poor water control and cell shape control. It is also said to be electrogenic because it contributes to the function of the nervous system as well as the muscles. So what we see here is that the first step is that sodium binds to the sodium-potassium pump. The next is that the sodium binding stimulates the conversion of ATP into ADP, and so there is energy release that moves the sodium into the cell. Why? Because the channel changes shape and literally spits sodium out of the cell. This change in shape also tends to allow the binding of potassium from outside of the cell, and then there is loss of the phosphate and restoration of the protein to original shape. And when this happens, the potassium is literally spat into the inside of the cell, into the intracellular fluid, and the cycle continues. So what we see is that 3 sodium particles will be pumped out of the cell and 2 potassium particles will be pumped into the cell. Therefore, we end up having sodium as the most abundant electrolyte outside the cell and potassium as the most abundant electrolyte inside the cells. Then we look at secondary active transport. What we see is that secondary active transport, one chemical substance is pumped using ATP and creates a gradient which allows that chemical substance now to begin to diffuse using another channel. Now the diffusion of that substance uses to open the movement of a particular solute against its concentration gradient. Here we are illustrating that, for example, hydrogen ions are pumped using ATP through proton pump into this one side of the cell, where now the diffusion of hydrogen ions back will actually move sucrose into a concentrated environment. This is called secondary active transport. So the ATP is used in the first step, but the diffusion is what moves the other substance against its concentration gradient. Okay, so these are illustrations. Then finally we talk about bulk transport. So this is another subtype of mechanisms of transportation where molecules are transported across the cell membrane without literally the substances passing through directly the cell membrane. So it's like the cell membrane is either swallowing or literally spitting the chemical substances. So we have 2 mechanisms. We have endocytosis or exocytosis. Endocytosis, we have 3 types. We have phagocytosis, which basically involves the taking up of solid substances into the cell. Pinocytosis, which is like taking substances which are in solution, and then receptor-mediated endocytosis for specific special substances that uses receptor for the cell to be able to take those substances in. And so these are illustrations for phagocytosis, and then it illustrates also that special cells like white blood cells tend to use phagocytosis as the mechanism of endocytosis to actually perform their differentiated function. Then we have receptor-mediated endocytosis, which is very, very important also for special substances like vitamins, which are recognized by particular cell membranes, and these are taken into the cell as a process of endocytosis. The process of exocytosis is useful for particular cells like um, glands that produce hormones, glands that produce exocrine secretions, and also neurons that produce neurotransmitters. So when these substances are produced within the cell, they are contained in a vesicle, and the vesicle is moved towards the cell membrane, fuses with the cell membrane, and literally opens up to spit the rest of the substances that should be secreted. So this is the end of membrane transport basics. Study well. All the best.